Hi, my name is Song. My poster presentation today is regarding link RNA profiling and its potential usage as a thyroid cancer biomarker. I will talk a little bit about the background, and also I will talk about the whole workflow we generated from the database, the profiling of the samples, and the final biomarker generation. So people may want to ask, why make efforts on the thyroid cancer biomarker? Basically, it's all about the diagnostic challenge of the thyroid cancer. As you all know, thyroid cancer is a malignant disease originating from follicular or parafollicular thyroid cells. The first symptom usually is the nodule of the thyroid region. But the challenge here is the thyroid nodule is pretty common in the whole population. It could be as high as 67%. So the identification of the malignant thyroid nodule is critical to the clinical diagnosis. That's why the researchers keep working on it for a better diagnostic tool. Our group, I will work with Cornell University, Dr. Ficki's group for messenger RNA-based biomarker development, and our effort is keep working on. Recently, long non-coding RNA got into the view of researchers for its important role in the biology and the pathology. Several long non-coding RNAs have been linked to cancer development as either the oncogene or the tumor suppressor. And link RNA-based biomarker have been suggested by several different publications. That's why we want to generate a link RNA-based thyroid cancer biomarker. For the link RNA-related study, the first difficult part is the database. Currently, there is no database that can cover all the known link RNAs, especially for those well-characterized link RNAs. QIAGEN will make our effort to merge two important databases together. One is the RAF-seq database, one is the Ensemble gene code database. So we generate a full transcriptome database. This database covers most of the annotated link RNA. With that database, we can design link RNA assay with high sensitivity and specificity. So that makes us have a solid base for the development of link RNA array. For the thyroid cancer biomarker development, because the lack of the thyroid-specific link RNA information, we decide to use our cancer-related link RNA PCR array for the thyroid tissue sample screening. This long non-coding RNA PCR array that generated with 84 link RNA target well selected based on the peer-reviewed paper. It also contains a panel of controls that include the housekeeping genes, the genome DNA controls, the reverse transcription controls, and the qPCR efficiency controls. Those 84 targets include the link RNAs that have the correlation with the diagnosis, staging, progression, and the prognosis of cancer. It also includes the link RNAs that have a role as a tumor suppressor or the oncogenes. So we believe with this link RNA array, we can identify the thyroid cancer specific biomarker. With this cancer-related link RNA array, the whole workflow from sample to result is pretty easy. The thyroid cancer samples and the control samples was processed to generate the purified RNA, and then the RNA was converted to cDNA with our first strand kit. Then the reaction mix was applied to the array and run it with the qPCR cycler. The raw CT were further processed and get into this random forest-based biomarker program. By default, we generated a 16 link RNA panel that include PTCSC1, which is a link RNA have been linked to the thyroid cancer that have a 3.4-fold change. It also includes the BNSCR that have a 3-fold change and SNHG16 that have 2-fold changes. After we got this 16-gene panel, we want to know whether it have the potential to be used as a biomarker for the malignant thyroid nodule identification. So we did a cross-validation. The result is encouraging. You can see here the area under the RC curve is 0.99 and accuracy is 0.96, while the true positive rate is 100%. If you take a look for this class probability figure, you can see that the positive one or the malignant one, here the red one or the benign one, can be classified clearly. And if you set this 0.5 as a threshold, you can see all the malignant nodules can be classified as positive. If you take a look for this heat map, you can see the clear discrimination pattern of these two known groups. The red is the control, the blue is the cancer panel. So do you have any questions?
How many long non-coding RNA assays do you have now?
Currently, we have more than 60,000 lincRNA assays that cover human and mouse. In general, it will cover not only the lincRNA gene, but also the lincRNA transcript of a coding gene.
Would you be able to generate lincRNA assays based on my own list?
Sure, we definitely can generate custom array based on customer's gene list. It is better to use the gene from the RAF-seq or the ensemble.
Is it possible to add some mRNA to long non-coding RNA array panels?
Yes, definitely yes. So we have two choices. One is you can pick 1 to 4 messenger RNA assay and add it to the cataloged lincRNA array that we call the modification lincRNA array. You also can generate full custom array. That means you pull 2 different kind of assays together that include the messenger RNA assay and the lincRNA assay. We have the full flexibility.